Thank you. All right, welcome back. We are going to be going over muscle and muscle tissue in this lecture. This is a pretty in-depth lecture, so if there's any questions whatsoever, pause the tape, write down your questions, try to look them up in your lecture book first, and then contact me if it doesn't make sense, either through email or a phone call, or contact me during my office hours, okay? So muscle and muscle tissue. The actual named muscles we'll learn in lab. We'll actually be doing them with clay on models in lab. We'll learn them. We'll learn their beginning, their ends, and how they function and how they're structured, okay? So the actual named muscles you do need to know for lecture, but we're gonna learn them in lab, okay? Muscle tissue, we're gonna talk about in lecture, okay? So overview of muscle tissues. There are 3 groupings of muscle tissues that we've reviewed in the past. We're going to review again. Okay. Those 3 groupings consist of skeletal. Let me see if that's coming up. Good. Cardiac and smooth. So when we talk about the musculoskeletal system and voluntary contractions, we'll be talking about skeletal. So skeletal muscle will be attached to bones. Cardiac will be found in the heart. Smooth is going to be found in visceral organs or viscera. Okay, such as the respiratory tract, the GI tract, the uterus, okay, attached to rectus pili, or they are rectus pili attached to the base of hairs, okay. So smooth we'll find in hollow organs for the most part, okay, and other areas of involuntary contraction. So these are the areas that they're located. Let's talk about how they look microscopically, okay. When we look at skeletal muscle underneath the microscope, it's actually what's called a myofibril, okay? So the smallest functional unit that we're talking about in skeletal muscle will be a myofibril. Actually, the structure's inside of that, but the cell of skeletal muscle will be called a myofibril. And what it looks like is going to be like a long tube. So this is just a section of it. It's very different from our typical cell that we've looked at, the composite cells in the past, where we have the round cell with the nucleus in the center. It's not like that. It's a nice, long, thin fiber. And there are multiple fibers stacked together to create the muscle itself. Now, skeletal muscle, just one individual fiber, when we look at it microscopically, will have what are called striations running through it. Okay. I'll explain to you why that happens in just a minute. And the other microscopic characteristic is that it is multinucleate. Okay. So it's striated, which means that it has altering dark and light areas, and it's multinucleated, which means there are multiple nuclei within that cell. Okay. Cardiac is also striated. The cells are much shorter and they have a Y-shaped appearance. So they are striated as well. They have a Y-shaped appearance and they don't have multiple nuclei, they only have one. Okay. This cardiac cell talks to 2 more, which talks to 4 more, which talks to 16 more, which is exponential. That way we can move that electrical signal through the heart quickly and efficiently. Okay. Finally, we have smooth muscle cells. And smooth muscle cells are going to be pointy on both ends, and they're smooth. They have no striations on the inside. They're mononucleate, which means they have one nucleus right in the center. Almost looks like an eye. Okay. So this is how they look microscopically. What we're really going to focus on looking at microscopically is going to be skeletal muscle. Well, we've looked at all 3 of these as a general example. What I mean is specifically we're going to look at all the parts that are inside of skeletal muscle. Okay. Deep inside the skeletal muscle, so this would be one cell, inside of this are teeny tiny fibers, millions of them, called actin and myosin. They're called myofilaments. And what those myofilaments do is they overlap. Okay? There's a thick fiber and there's a thin fiber. We're going to get into this in a little bit. Okay? The thick fiber has little levers on it, many, many little levers. And what happens is when this muscle is stimulated, that thick filament's levers attach to the thin filament and slide it a little bit. Then they release and they slide a little bit more and continue down. And you can see how when that happens, the length between my arms shorten. The same thing happens with those little filaments in skeletal muscle. They shorten the length of the muscle. And then those little levers help to unwind that muscle or let it lengthen out, okay? That's how these little fibers overlap. And you can see that there are areas that these fibers overlap. Where these areas overlap is where we get the darker areas on the striations of skeletal muscles, and where they don't, there's a thinner area that looks lighter. Okay, so we get that alteration of dark and light because of those fibers that are inside the muscle cell. Okay, myo-mys and sarco are all prefixes for muscle. Okay, so skeletal muscle, if you go to your next slide, we've gone over most of that. They have striations. They are voluntary. All skeletal muscle is voluntary. Okay, so we have conscious control over it. And it tires easily. It tires easily because it's very metabolic. And being very metabolic means it uses a lot of energy. That energy, don't forget, is going to be glucose and oxygen feeding into the cells and creating our storehouse of energy called ATP, or adenosine triphosphate. Skeletal muscles use a ton of ATP. Cardiac muscle, once again, heart, it's striated, but it's not voluntary. We have some ability to control heart rate just due to levels of stress or relaxation, and also our level of activity, okay, can help to change the heartbeat, but the heart beats regularly on an involuntary basis. Okay, smooth muscle is going to be in the hollow organs, okay, areas of involuntary contraction. Helps to force fluids through, helps to regulate air going into the lungs, does not have striations, and it is also involuntary. Okay, so functional characteristics. I think excitability is pretty common sense. Okay, obviously muscles will be excited. They will be excited through a process called an action potential, which we're going to talk about in a little bit. If you remember from A&P 1, an action potential is the electrical activity that makes a nerve or a muscle work. So all of these muscles are stimulated or excited through that action potential. That is the excitability component of this. They all contract and shorten. When a muscle is stimulated, it shortens, okay? If it's in a resting state and it is stimulated, it won't stimulate and lengthen, okay? It will not stimulate and lengthen. You can't think about your hamstrings and think, well, I'm going to force it to stretch with your movements. Doesn't happen. The only way that you can extend or lengthen a muscle is passively through stretching. If you excite a muscle, it will contract and shorten. Okay? They are extensible. You can extend them and stretch them. When you extend them and stretch them, they don't stay at that length. They will recoil back to their original shape. Okay? They'll be a little bit longer if you keep stretching. You will get more flexible, but for the most part, they'll resume back to almost their original shape. Okay. Muscle functions, so movement's an obvious thing. Posture and tonicity, this is an important concept. There are areas in the body that muscles have a constant slight contraction. And that constant slight contraction keeps them in a state of readiness to be able to contract, okay? It's called tonicity. This is an important concept in the GI tract because the GI tract needs to be ready to take in foodstuffs and digest it and excrete it. And this concept is also important for postural muscles. When you're moving through space, the muscles that keep us upright are in a constant state of slight contraction, and that helps to regulate us so we can maintain our balance in space. Okay? Muscles also stabilize joints. Very important component we've talked about when we talked about joints last week is the muscles crossing the joint help to stabilize it, especially in the rotator cuff. Okay, rotator cuff is only held onto the skeleton at the sternoclavicular joint, so the muscles of the rotator cuff help to hold that shoulder joint in place and to generate heat. One of the byproducts of all the biochemical reactions in muscles is the release of heat. It keeps us warm. Okay, skeletal muscles, keep on moving. So each skeletal muscle has one nerve and one artery and one vein. Okay, that artery and vein, or I'm sorry, artery and nerve and vein actually would enter the muscle at one area, and then that would divide, that neurovascular bundle would divide down, so every muscle cell has a nerve, artery, and vein going to it. And if it didn't, it couldn't contract, it couldn't get blood supply, and it couldn't drain its waste. Okay, so even though there is one going to a muscle, one nerve, artery, and vein, it will divide many, many times until each muscle cell has a nerve, artery, and vein. Okay. Once again, skeletal muscle needs tons of energy. It's very metabolic. It's actually very selfish with its glucose stores. And if you remember, glucose is stored as glycogen. Skeletal muscles are very selfish with their glycogen stores. They don't share them. They keep them to themselves because they need this constant, quick supply of energy. Okay, connective tissue sheaths. If you take a look at this, I'm going to zoom in on the PowerPoint. This picture is right in your book. I know this might be a little bit difficult to see. So if it is, go ahead and pause the tape, open up your book to this picture. These are the connective tissue sheaths that encase the muscle and connect the muscle to a tendon which connects to the bone. And this is the breakdown of the names of those different connective tissue sheaths, okay? If you look at the outside, that's the epi, which means on top of mesonym, okay? The epimysium is going to be a dense irregular connective tissue around the entire outside of the muscle, okay? That's going to create a little bit of an anchor for the outside of the muscle as it connects to that tendon. The perimysium creates fascicles. If you look at the little bunching of different muscle cells that are bunched into little groups, those are fascicles. The perimysium is going to be the connective tissue encasing those fascicles. The epimysium is going to be the connective tissue that surrounds each muscle fiber, and each muscle fiber is actually a muscle cell. I really want you to take a look at that. You can see the big muscle and how it's comprised of all those muscle cells. Those muscle cells are grouped into those fascicles. There are multiple fascicles which make up the muscle itself. If we look at that muscle cell, the epimysium is basically going to be the cell membrane for that muscle cell, okay? You can see it looks like a nice long tube that's full of fibers. And those fibers are those teeny tiny fibers that overlap, that shorten the whole muscle that we talked about earlier, okay? So I want you to visualize that muscle cell. Imagine that I'm taking that little muscle cell out, that big long tube, and I want to show you the inside of that muscle cell. Okay, which we'll do that in the next couple of slides. We'll review the inside of that. Okay, so the attachments. Okay, people always get confused about the attachments of the muscles. Okay, don't overthink this one. The attachment sites of the muscles are simply the areas to which they are attached. The first concept that's important to understand is that in order for a muscle to work, it has to cross a joint. For example, the bicipital muscle. Okay, the biceps is going to actually start in the scapula, and it's going to insert down onto the radius. And what I think most of us know the biceps does is to flex the forearm. If the bicipital muscle only attached onto the humerus, the beginning and the end of that muscle attached on the humerus, and it tried to contract, it wouldn't do anything. So the first important concept is that muscles have to cross a joint in order to move the bones around that joint. Okay? So the attachments. There's an origin and there's an insertion. These are the names of the ends of that muscle. Okay, there's an origin, there's an insertion. The origin is the beginning of the muscle. Okay? It tends to be more proximal to the body. So if with the biceps, the origin for the biceps is here on the scapula. The insertion would be more distal. It attaches onto the radius. It's named as such because that is how the ends of the muscles are named. The origin is more proximal. The insertion is more distal. Okay? The origin in muscles is fixed. It doesn't move. The primary movement for biceps is to flex the elbow. Okay? So my shoulder doesn't move when my bicipital muscle is working. Okay? Biceps brachii, I should say. Okay? It stays in place and my elbow moves. So the origin is fixed. The insertion is the part that moves. Okay, so origin and insertion are the two ends of the muscle. Origin is more proximal, insertion is more distal. Origin is fixed, insertion moves. Now there are some muscles that don't run up and down on the body. There are some muscles which run horizontally. Okay, for example, part of the pectoralis muscle runs in this horizontal fashion. Okay? The origin in that case would be medial. The insertion would be lateral. So the origin for the pectoralis muscle, this region of it, because there's different regions of it, okay, would be, the origin would be medial, the insertion would be lateral, and what would happen with movement with the pectoralis muscle would be moving the shoulder in. So this end would stay stable or fixed. This end would move. Okay? Origin, insertion on a horizontal muscle. Origin, insertion on a vertical muscle. Okay? Now, for the most part, muscles are going to insert onto a connective tissue. For the most part, it's going to be a tendon. A tendon is going to be a nice focused insertion for that muscle of really regular fibers running in one direction. And aponeurosis is a little less common, and that's going to be dense irregular connective tissue going multiple ways. Okay? So attachments, the two attachment sites for muscles. Okay? So now we're going back to the muscle cell, okay? That teeny tiny tube that we looked at in the picture 2 slides ago. We're going to look at the inside of that. So if you can imagine that teeny tiny tube is full of millions of fibers. And those fibers are overlapping once again and they shorten, which we talked about before. I'd like to show you more specifically the parts of those fibers or those different types of fibers. Okay? If we think of we're just going to look at this skeletal muscle for now. So here is that tube. Okay, here is that tube. The outside of that tube is going to be a cell membrane. Okay, in muscles, it has a specific name. That cell membrane is the sarcolemma. Okay. So it's simply the cell membrane. Somebody, of course, wanted to mix this up and give it a new name, and they called it sarcolemma. Okay? That's going to be the cell membrane for a muscle cell. Inside is going to be the sarcoplasm. Once again, it's a more specific name for the cytoplasm, or the gel-like substance that's inside the muscle cell. Okay, so sarcolemma is going to be the cell membrane. Sarcoplasm is going to be the cytoplasm inside of a muscle cell. Okay, and of course we have those alternating striations. Now, inside that sarcoplasm, we're going to have millions of overlapping fibers. Okay, they're called myofibrils. They're actin and myosin. They're very structured in very symmetrical patterns inside the muscle cell. Pardon me. Okay, what I would like to do is show you one little section of those myofilaments to show you how they overlap. Okay, so imagine there are millions in there. I'm just going to show you a little section of how those fibers overlap. So I'm going to go to the PowerPoint slide. Okay, this should be in your notes and it's also right in your book. So if you can't read the words, once again, get your book open to this page, please. You can see the blue and the red. Okay? The blue is the thin filament called actin. The red is myosin, which is also known as a thick filament because it is indeed thicker, okay? And the actin is thinner. Now, there are attachment sites on actin that are usually covered up. And those attachment sites, once they are exposed, those little levers on the myosin will attach to the actin and slide those actin filaments across it. The actin filaments will actually get closer. You can see there are two of them. They get closer to each other and they shorten the length of that muscle. Okay? So the myosin, or the thick filament, is going to latch onto those attachment sites on the actin and slide the actin towards itself, shortening the full length of the muscle. Okay? The primary ion that's in charge of that, and it goes more in depth than this, but I'm trying to keep it a little bit simple for you. The ion that's in charge of that is calcium. Okay? Calcium exposes those binding sites on actin so myosin can attach to it and shorten the length of the muscle. Okay? You can see the parts of the myofilaments, okay? What you need to know is from the Z disc to the Z disc. I'm not going to make you memorize all the different parts, but the smallest contractile part of a muscle cell is called a sarcomere, okay? It's called a sarcomere. The sliding filament theory is that theory I just explained to you about myosin grabbing onto actin and sliding it across itself to shorten the muscle, okay? Sarcoplasmic reticulum. Now, I just mentioned that calcium is going to be the important ion that will allow those filaments to contract, okay? The sarcoplasmic reticulum is going to be the structure that stores the calcium, okay, and releases it when a muscle cell is stimulated so it can cause that contraction or that shortening of the myofilaments, or the actin and myosin. Okay, so it's stored in the sarcoplasmic reticulum. The terminal cisternae is going to be, I'm not really going to get into that. Okay, the terminal cisternae is going to be sort of like the ends of the sarcoplasmic reticulum that's very close to these little in-depth indentations called T-tubules. Okay, we're just not going to get into that. What to keep in mind is those two structures help to facilitate the release of calcium throughout the muscle cell quickly and efficiently. Okay? So what you need to know off this slide is the sarcoplasmic reticulum is going to store and release calcium, which will cause a contraction of muscle. That is what you need to take from this slide. Okay? Physiology of a skeletal muscle fiber. I'm going to pause the tape and then we will resume.